pick this up. But he's a very interesting person. He got a PhD in physics from Harvard a number of years ago now, and he did a lot of instrumental work in early computer science, before there really was a field of computer science, and computer networking. And it turns out the biological signaling involved in health and aging is very much like a computer network. Lots of interacting pieces that interact in oftentimes unusual ways. And so he's had a long time as kind of a hobby of the mechanisms of aging. But about 7 years ago, he really got serious about it. And he's been writing a blog, agingsciences.com, that he now has 450 articles in, and it gets more penetrating and more interesting all the time. So I don't think I'll spend any more time introducing. I'll give you Dr. Giuliano.
Do we turn the lights down?
Oh, yes. Sorry, you're right.
I am very honored to be here and have this opportunity to talk about a subject that I'm very passionate about, which is aging science, what we know, and a tremendously interesting and evolving field. I'm very grateful for Dr. Drafts for having originated this invitation, and I'm just delighted to be here. I will try and keep myself down to 45 or 50 minutes, which is very, very difficult for me to do, and then have some time for interaction and comments and questions.
First comment, we'll hope to have this presentation up on our webpage, and hopefully all the technology goes right.
Some of the topics I'm going to talk about are state of the sciences of aging. First of all, aging isn't a science, it's a whole set of sciences. It turns out that practically every life science in every field of medicine impacts on aging. I'm going to talk about some important things we've learned about aging, the consequences of aging, diseases, health spans, what to do or what could be done about extending human lives, why that might be something very important to do socially, and something about practical steps for living 10 or 20 years longer. Some of these are Dr. Oz type conventional wisdom, and some of them are not so conventional wisdom, and some of them are controversial. And secondly, about breaking through the 122-year age limit. As far as we know in history, there is no documented example of somebody living more than 122 years. What is the possibility of extending life beyond that, of having people be healthy for 150 years or 200 years? And what might be a technique that makes that possible? So start out, first comment is that the state of the scientific literature bearing on aging is vast. We're looking at more than 20 million publications, something like 21 million publications, 1 million new publications coming in a year, having to do with the life sciences. And by gosh, if you look for the word aging in those articles, they are spread across. They're in the cancer literature, they're in the aging sciences literature, they're in the Alzheimer's literature, they're in the literature having to do with pathways. They're all over the place. So it's a vast field, and it takes us into everything. Well, what do we know about the sciences of aging? I hope you all understand this diagram. It's an example, one example of many complex biological diagrams that are used to characterize the field of aging, or used to characterize biological processes. The one on the left is a simplified diagram of metabolic pathways. I'm sure Professor Dratz could find many more complicated diagrams. The ones on the right have to do with purine metabolism. So there's hundreds or thousands of such diagrams that keep appearing in the literature. They are complicated. They are deep. And the research is solid, and I'm grateful for the research because it informs my work. I could certainly not begin to pursue the kind of science involved. But it's vast and deep, and the researchers in one field who may produce one diagram, if you go on Google Images and you search under, say, you search for metabolic pathways, you'll find 20 diagrams or 40 diagrams, and everyone will be different. So that we're characterized by different fields of research who pursue different theories, they pursue different ways of looking at things, and the results are disparate. Many scientists are only partially aware of what other scientists are doing because of this depth and what we call the siloing of knowledge. And so, therefore, what I'm going to talk about is my story about aging. And if you had another 15 aging scientists and had them give them their stories would be different. My story is based on what I do, which is survey the literature. I spend 40 to untold numbers of hours every week going through the latest publications, picking a topic, trying to summarize what's known about it, interacting with colleagues about it, drafting documents, having those documents reviewed, trying to make sure I really understand what's going on, and then publishing it in my blog, agingsciences.com. So I don't have any vested interest in any one theory, but rather, and nor do I have the real depth of research that many real researchers have in many of these areas, but I cut across a lot of areas, and I have a view. One way I have described the situation is that I fly 15,000 feet above the biomedical literature, and then every once in a while I dive down to maybe 30 feet and do an in-depth study of some field. So a way of seeing how the situation is today is based on jigsaw puzzles. How many here have done big jigsaw puzzles? Really big jigsaw puzzles? 500 pieces, 1,000 pieces, 1,500 pieces? Okay. Then imagine that we're putting together a jigsaw puzzle of very large size. I don't know, we don't know the size, maybe 30 million pieces. And imagine that there's no box cover that's going to tell us what it looks like. That's kind of the situation we're in. And oh yeah, it's not just a two-dimensional puzzle, and it's not even a three-dimensional puzzle. It's a puzzle of very high dimensionality. So that's the kind of situation we're in. And where we are is like where we are when building a jigsaw puzzle. You put together the pieces of edge that seem to fit together, and you get a chunk of edge and another chunk of edge, and then you get a chunk of what you think may be sky or maybe it's water, and you get some images of the farm building, but maybe they're the castle, you know, and then you build those, and then you can connect one big chunk with another big chunk, and pretty soon you have got the puzzle solved. And we're in the midst of that process. We've got a lot of the chunks that enable us to understand a lot of things about aging, but certainly not everything. And we're in a situation where it may well be the case that the puzzle will be coming together soon. At least there's excitement to believe that the knowledge is coming together and that we'll soon have a better grasp of aging. Now, this diagram is a very interesting one because one of the things you learn about aging is that we're living longer. Now, we're living longer, that is, on the average. Whoop, I went ahead too far here. Go back. This curve on the left here represents the situation in about 1900 of how people survived. This curve over here between the red and the green represents today. And you can see that on the average, we're living a lot longer. The average lifespan from birth of Americans is increasing about 3 months or 4 months every calendar year. That's how much we're living longer. So something is going on evolutionarily speaking, and I'll comment about that. That means that our lifespans are becoming longer. And this chart shows that how it's anticipated to look in 2060. And one way of saying this is that the survival curve is becoming rectangular, and if it continues going this way, we'll all live through 122 in the day and then drop dead. Which is not such a bad idea, by the way. Not such a bad idea on the whole. But at present, we don't know how to break through that 122-year limit. There is something biological, something programmed that means that this is the limit of aging as of the present. So first consequence of aging, it's fatal. But it's the best thing we have. So we know a lot of other things about the consequences of aging. Lots of things happen that are degenerative, declines we don't like, markers in the body, processes that run downhill, lessen immune responses, et cetera, lessen DNA repair capabilities. Things get less efficient. The DNA gets methyl groups attached to it, which means that its expression is less. Histones, which are the spindles around which the DNA is wrapped, get deacetylated, which means that the expression of genes is decreased. Key health genes, their activation is decreased. The activation of cancer and other dangerous genes is raised. There's more and more DNA damage. The list goes on and on. This is not a very pleasant list, but you know, it's really incremental decrepitude. Well, that's what we're facing. So with this decrepitude, there's a great acceleration later in life of the diseases of aging. This shows something about occurrences of Alzheimer's disease. You see there's an elbow there. Whoop, I'm going back, going too far again. You see there's an elbow in the curve there where it shoots up with advanced age. And the pattern appears with other diseases as well. These are malignant cancers. And again, we see how they shoot up more for male than for females, interestingly enough. With these diseases, we find that the aged consume many more healthcare resources. Oop. I'm looking for the pointer here. Okay, I'll learn this. I will. This curve is for the United States. These curves are for other countries, so you can see how out of whack and out of control our healthcare costs are per capita for aged people in the United States compared to other countries. It's really ridiculous, but you can also see how much of the healthcare budget is consumed by aging people. And the other slides reinforce this. This one here shows cost of fall injuries by age groups. And it's a worldwide trend. You can see that the cost of aging and the cost of age-related diseases, this is for people with dementia, is worldwide. So it's a worldwide phenomenon, particularly even though their healthcare costs are less than ours in other countries. What we're looking at is a phenomenon that's quite ubiquitous. So, okay, what about extending human lifespans and health spans? There are multiple studies of aging, a series of aging. There's a huge stream of new discoveries that bear on aging. You look at the number of papers that, publications that come out that have a statement to the effect of, we believe these consequences have significant impact on the understanding of aging. Well, you know, out of the 1 million new publications a year, perhaps a third have that statement in them. So what do we know about extending health spans? Actually, it's an easier matter to grapple with than understanding aging itself, because to understand aging, we have to understand essentially all of human biology. This is extremely complex, extremely deep. But it may be that understanding how to obtain certain number of additional years of life, or average life, is a much easier issue. I believe it's a much easier issue. Well, we know how to slow aging in lower animals. Like there's aging interventions that worked in nematodes, fruit flies, and mice. And we can just about double the life of a mice. We can multiply the life of a lifespan of a nematode by a factor of about 8. This is very impressive. But many of the interventions used to do these things are not practical for humans because they involve messing with the genome, genetic interventions. And when it comes to human beings, we just don't mess with our genomes right now. We're willing to try other things, but we're not going to do that. So these interventions give us a lot of insight as to what's going on in aging, but they're not necessarily ones that we can apply directly and simply. One important observation based on animal experiments is that the lifespans and health spans are very closely correlated. You create an intervention that extends the life of a nematode by a factor of 6 or 8, and they still die of the same causes, but those causes occur much later in their lifespan. You double the life of a mouse, still dies of the same causes, but those causes occur later in the lifespan. So that we have a perspective that lifespans and health spans are very closely correlated. And so this leads us to a different way of thinking about degenerative diseases. For a long time, degenerative diseases were thought to be, aging was thought to be a risk factor for degenerative diseases. Things like Alzheimer's disease, cancers, diabetes, the things that kill older people. Now, we're beginning to say, well, aging is a causal factor for those diseases. You know, aging causes it. Now, what does that mean? That's just another way of expressing our ignorance of what those causal factors are. But it leads to a suggestion that there could be a shift in research towards, instead of trying to cure cancers, here we are 40 years into the war against cancers. We spend, I don't know how much in aggregate, many billions, perhaps trillions of dollars on cancer research and research in Alzheimer's. And it suggests that maybe what we should try to do is address aging as a way of postponing those diseases. And this would make a lot of sense given the really very low productivity and poor results of most of those studies. We still really don't know how to cure Alzheimer's disease or Parkinson's disease. We still don't know how to cure more cancers, and the breakthroughs that are reported in the press are often very partial breakthroughs. That what got me into this field was realizing that reading newspaper reports that somebody has solved this problem. They made a big breakthrough, and it's reported in all the press. And usually it's a result of a university release of some professor's work. And usually when you look at it, you find that yes, this could be an important incremental piece of research, but actually there's a whole stream of research that goes with it. And that stream is not done. Now, this slide talks about what's at stake here. Well, suppose that we could just simply add 10 years to the average age and the average health span. Only 10 years, okay? That means that the healthcare costs of this person, which is at this point on the curve, would go down to this point. The savings just in healthcare costs, not even to mention the savings of increased productivity, the benefits of having senior people be able to contribute their wisdom to go on, would be immense, just immense, from only just 10 years of gain. I did some calculations at some point that said that if we could get this benefit on the average, if we sped up that process that's going on already about adding to age spans and could move things up 10 years, the economic benefits would more than offset the vaunted budgetary deficit, the vaunted lack of productivity. And I feel, personally, that this century, having lived through and contributed to the computer revolution in the last century, I personally feel this century belongs to biotechnology. It belongs to the health sciences. It belongs to aging. And that if I can manage to live that long, if I can manage to keep ahead of the Grim Reaper, who's just 3 feet behind me right now, if I can keep ahead of him, I will see this benefit happen. Again, I point out another thing going back to this curve here, that that's where we were when the Social Security Act was passed in 1935, and the average U.S. life expectancy was 64.5 years. And here's where we are today, where the average U.S. life expectancy is 80.5 years. A lot of people are living to be 100 years now. Could we move it up to 10 years? My feeling is no problem. Well, there is a no problem scientifically. Socially, it would require a massive engineering factor. So I see the extending lifespan divides into really two kinds of questions. First of all, what can we do to add 10 or 20 years? What can we do to get that 10 years or to add 20 years on the average? And of course it's statistical. There's still people who are going to die younger, or there's still people going to die older, but we're moving the curve up. We're going to have more of us living longer in the lifespans that we know we can achieve right now, because there's a lot of people living them. There's a lot of centenarians now. Great acceleration. The centenarians is the fastest growing percentage-wise demographic group. Weird, but that's what's so. So the second problem, second issue, I think is a really different issue, and it requires a different technology and a different approach, is what can we do to break through the 122-year limit? This means we've got to break some very basic rules in history. This means we have to go beyond what we know to be impossible now. But what can we do to extend lifespans to make living 150 years possible? Or maybe 100, 200 years, or 500 years? Nobody knows where the limits are. But I'm going to talk a little bit about both of these. And first of all, about the additional 10 to 20 years. Well, in my opinion, this means having more of the people who live longer right now and fewer of the people who live shorter right now. And applying, I believe, believe this can be done applying conventional wisdom about health and some unconventional wisdom. I believe this is entirely possible. Genetics, well, there's the idea of, okay, we have our genes and we're stuck with our genes, and because we're stuck with our genes, we're going to die when our genes say we're going to die. This was a viewpoint. Now, how many believe this is so? Okay, some people believe this is so. I think we now know that with the exception of certain rare, completely genetically driven diseases, that a relatively small proportion of disease incidences is driven by having genes. So you have pro-cancer genes. Okay, if you, through lifestyle and other interventions, that doesn't mean that means you can still, I believe, reduce the probability of you contracting a cancer. I think you are not doomed. I believe that there are actions that one can take to defend themselves against most of the SNPs, the gene polymorphisms that have disease susceptibilities. So I do not see genetics as being a driving factor here. It's something else. I believe it can be done by paying attention to these things, and I'll talk a little bit about these and a little bit about practical things that I do and I see that's possible using them. And there's also pharmaceuticals in the pipeline now that could extend life. People would like to take a pill and live longer. Well, there are such pills that are being researched and being developed. They're not being developed to cure aging because aging is not an FDA indication. You can't go to the FDA and say, I want to cure aging, because they say, I'm sorry, they look at the list of disease and aging is not there. But many of the drugs being developed for diabetes, Alzheimer's, and cancers might be applicable. Okay. So I believe it's a personal choice. I think there's enough known now that as individuals, if we're willing to learn what's known and take good advantage of it, and I believe there is evidence for this in the literature. I believe this is an evidence-based statement. It's possible to be one of those who lives longer instead of being one of those who lives shorter. So I think getting that 10 years, I've done it. You can do it. It's not rocket science. It's possible. It's entirely feasible. But not everybody knows they have that choice. And even those who know they have the choice may elect to not want to disturb their lifestyle or see the task is overwhelming to do that. Or if you're younger, you may feel, well, I've got a long ways to go and I don't need to worry about aging right now. I'll worry about it later. That's what I did. So I believe that picking the longer lifespan, picking another 10, 20 years, requires some learning, following some rules of discipline, and that it's entirely feasible. Now, I mentioned that your genes are not determinative, but which genes get turned on when and how is very, very important. There's a new field called epigenetics that has to do with what turns the genes on and what turns them off. When we had the human genome process, we thought, well, not very long ago, we thought that once we figured out all the genes we have in the DNA, we know that the genes make the proteins, and the proteins is what keeps us going, and that we would understand what life is all about and what we are all about and how to cure diseases. Well, the only problem with this is that it was wrong. And where it was wrong, this is a very important step. I mean, I'm glad we had the step. We had to have that step. But where it's wrong is that what's really important is what turns the genes on, what turns them off. If every cell in your body has the same genes, well, how come one's a brain cell and a liver cell and one's a skin cell? You know, there's a lot of difference between your cells. And you've had the same cells all your life. So what's the difference between a young cell and an old cell? What's the difference between one cell in your body and another cell? It has to do with gene expression, which is this whole idea of epigenetics, which means really beyond genetics. Okay, these are the points I just made. Now, gene activation patterns do change through life and with aging. And so one of the key areas of longevity research, I believe, that's apt to be productive is the whole field of epigenetics. We decoded the genome. We have only scratched the surface of beginning to decode the epigenome. I'll talk a little bit more about this later. The epigenetic information, how is that encoded? Where does it show up? Where do we find it? Well, at one point, we had a gene over here. We have a strand of DNA, okay, a chromosome, a strand of DNA, and you find a gene. And then lots of stuff, we're not sure what that stuff is. We're going to call it junk DNA. That's what we call it, junk DNA. And then another gene, and then a lot of other junk DNA, and then another gene. Well, that junk DNA is not junk. It actually determines what genes get turned on and off, and that junk DNA is something under your control. When you exercise, you influence what's in that junk DNA. When you eat something, you eat a steak or whatever you eat, or whatever your lifestyle patterns are, is encoded in that junk DNA, and you inherit that epigenome. Now, I grew up when it was a religious matter that the Russians believe, Lysenko believes that hereditary, that experience changes are not hereditary. This was a heresy of the Russian, but we geneticists know that you can't inherit experience. Well, that's wrong. You can't, and you do it because those parts of the DNA between the genes are malleable. They're flexible. They're the epigenetic markers, and they're inheritable. Okay, another way this information is encoded is in how genes are silenced by being methylated. They have a methyl chemical group, some attached to them, attached to their promoter, and that methyl group says to the gene, shut up, don't express, don't make protein. You just be quiet, be a good gene, but keep a low profile, all right? That's another way that methylated, that's being extensively studied. It's whole new fields of omics now, omics. You take a field of science and you add omics at the end of it and you have a new field of science. So there's methylomics, okay. I don't know how many omics. I think the last count was a couple hundred of them. Now, how tightly the DNA in the chromosomes is wrapped around the spindles, which are called chromosomes, is also affected by acetyl groups, which can be attached to those histones. And that's another way that this experience information is encoded. So it's encoded in several different ways. And this is all beyond the Human Genome Project. This is where the frontier of where things are going on is right now, as I see it. Whereas there's 25,000 genes or so, human genes, there are probably millions of epigenetic markers. We're only beginning to understand them now. So it's providing a whole new way. So what are the ways of living an additional 10, 20 years? Well, again, some of these are a matter of Dr. Oz type lifestyle, regular exercise, good rhythms of sleep and rest, a rich social, intellectual life. It helps if you have, if you're older, if you have companionship, if you keep active. there's many factors that have been studied, many interesting population studies. For example, one study shows that men who wives are still alive live longer. Another showed that polygamous men live longer than divorced men. I am not putting value judgment on these studies. And then 3 factors which I think are important: careful attention to diet, use of dietary supplements, and stress management. And in these last 3 factors, there's not only conventional wisdom, but there's some unconventional wisdom, and I'm going to try and relate a little of that to you in the process here. Okay? So conventional wisdom, you know, is a plant-based Mediterranean diet, fresh vegetables, fruits, and fish. Now, a guy probably who I most respect in terms of diet is Dr. Grass. And he eats meat. And he sees saturated fats are not necessarily bad for you. There's a lot of unconventional wisdom that's not in this conventional wisdom. Better, smaller meals, certainly paying attention to the omega-3 versus omega-6 balance. And the same advice is generally given for avoiding diabetes, insulin resistance, cancers, cardiological problems. So you find TV evangelists who have an anti-aging diet, or they have an anti-cancer diet, or they have, these are the guys who go on public radio on Vegathons, and you hear them, and they're usually pretty good, but it ends up, you look at their diets, they're pretty much the same when you come to the end of it. I'm going to talk very briefly about one pathway that I've studied, and I gave a more science-based lecture today where I covered this. There's a substance called NRF2, which is something that turns genes off and on. It's a gene transcription factor, and it has to do with what genes are activated. And it seems to be an important master regulator of cellular responses to certain stresses. And when turned on, it turns on some 240 health-related genes. And these genes protect you, turn on your own natural antioxidant response system, turn on detoxifying enzymes, turn on all kinds of defensive things. And you can get that by what you eat. One of the ways of turning it on, I mean, sure, you can, if you take metformin, or there's many chemical or biological substances that will turn this on, but also there's many foods that can prevent this. So, and in the process, make you healthier and prevent many disease processes. Now, there's a lot of literature on this. I've reviewed many of these substances in depth. I can't go into the details, but I want to say there's a science basis behind what I'm talking about. Here we've got some red peppers, which is capsicum. This is something called bitter melon, which is a folk remedy used throughout the world. It's a very important substance. Now, how many of you heard about bitter melon and its health benefits? Okay, there's something like 1,500 PubMed references to this, and we're looking at a substance that's embodied in folk medicine worldwide. Whether you go to the Amazon or Jamaica, where we've been doing some plant-based research work, or China or India, you find bitter melon. It's not very well known in the US. But you do know this one, which is ginger, and you've all heard about green leafy vegetables. Anybody here who's not heard about green leafy vegetables? So here are some of the common dietary substances that are known to activate NRF2. And there's a name and then where it occurs, you know. And here we find lots of things we know about, like chocolate, berries, blueberries, red grapes, onions, scallion, broccoli, these things that are good for you. And each of these substances has phytosubstances, and so the dietary use of phytosubstances, in my mind, is one of the interventions that can keep us younger. Now, they not only, whoa, what's that? What's that? What's that? It wants me to choose a profile. I don't want to choose a profile. Go away. And many of these dietary substances not only activate NRF2, but they have other epigenetic impacts. For example, they activate DNA repair genes, they inhibit inflammatory pathways. One of the theories of aging is that it's due to inflammation. Cancers are an inflammatory disease, and heart problems are inflammatory diseases. Controlling runaway inflammation is very important for older people. They change DNA conformation to make sure that the protective anti-cancer disease genes are expressed. That is, they keep the histones I was talking about deacetylated open so that the genes can be expressed. One recent article even argues that when you eat plant-based substances, some of the RNA becomes part of your own RNA, at least in the epithelial lining of the stomach, and perhaps elsewhere as well, so that the plants, which have natural anti-cancer defenses, plants, how many of you know plants getting cancer? Well, plants mostly don't get cancer. Why don't plants get cancers? They could. Why don't they? Well, they've developed very strong evolutionary defenses against cancers. I believe when we eat these substances, we are embodying the plant's defensive mechanism, things that have taken millions of years. Think of a plant as a little factory, a little biotech company that's taken a couple of million years developing a defense against cancer. And then imagine we eat the plant, we take advantage of that defense. It's kind of a simplistic view, but I think this is very similar to what plant-based substances do for us. They create stress signaling. I talked about hormesis, which is the study of stresses today at the scientific lecture. Well, then another area besides the diet and exercise is something about stress management. Now, how many of you think that this is correct? You want to avoid stress. Stress is bad. Yeah, stress is bad for you. We all know too much stress is bad. Well, again, this is kind of wrong. What you want is some level of stress, because we're human organisms, and biological organisms were evolved in the presence of stress. They're optimized to function in the presence of stress. And if we don't have stress, we're not functioning optimally. So we need stress. And you need and can benefit from stress. I talked about this again earlier today. But you need to manage the stresses and keep them within limits. The body of science involved is called Hormesis. You've all heard of this heavy-duty guy and his advice. I understand there's a popular song right now with this name. Again, organisms have evolved to function in the presence of stress. Hormesis is about that, and it's about how stresses impact on us. Stresses have a dual character at low doses. They can be good for us. And at high doses, they can be dangerous or they can kill us. So certainly avoiding excess stress. Exercise, how many exercises? Exercise. Okay, you're inducing stress. And as a result of you inducing that stress, a number of things are activated, channels that have been well studied, and you're healthier. You're less likely to get diseases. You're more likely to live longer than if you didn't do the stress. Now, how many have run at 7 miles an hour on the treadmill for 3 hours at a time? Okay, that's too much. The point is that there's a window of stress that's healthy. It's shown in this curve. This part below the line here is where there's a benefit to stress, and too much stress at this level, this is the amount of stress, gets you into damage. And there's a great deal to say about this. It applies to virtually all biological systems, whole organisms, and many, many interventions have been based that can increase health and longevity, certainly in smaller animals, used on the application of stress. And the NRF pathway I talked about is one of them. Again, some familiar examples of hormesis are these things. You induce stress by vaccines, and that makes you stronger, resistant against the pathogen. Grit training, you've all seen movies of the drill sergeant talking about chewing somebody out in their face and creating stress and say, give me 30 push-ups, That is toughness training. That, again, you subject soldiers or Boy Scouts to stress, and this occurs in many different forms in our society. Now, some of the important general properties of hormesis is that having the stress be within the window is important. Can't be for too long. It works best when it's synchronized with body rhythms, and you need periods of rest and recuperation. So, again, if you repeat stresses, like the more you exercise, the more exercise capacity you have. You want to accommodate to high altitude, then the more incidences you have of high altitude accommodation, the more you can do. You want to take advantage of cold shock stress. Well, the more cold plunges you have, the more you can handle the cold shock. So there's an accommodation effect. So in terms of personal health, the key is not avoiding stress, it's managing it. And this means welcoming the stresses of life. It also makes sure that they stay within your personal hormetic range, that they're not too much. Some of the stresses you can manage personally are heat and cold, exercise within limits, and endurance demanding activities, and lots of others. I used a slide earlier. In a typical day, I keep my bedroom pretty cold, and I'm very cold in the morning when I'm talking in the Boston area, Boston suburbs, and the temperature may be 58 or 60, and while I'm getting dressed and shaved and stuff for maybe 20 or 30 minutes, I'll be cold. So I'll be very, very cold. I'll take advantage of cold shock hormesis, which is a well-defined stress pathway. I'll eat these substances which take advantage of the NRF2 pathway. This is the person, this was on a typical day, and this person's mastery of English was wanting. My computer was misbehaving, and I needed help, and I couldn't get it. I had a little fight with Melody. I did some snow shoveling, which generated the PGC1 stress pathway. This is actually, Melody points out, this was a different day that week, but I spilled some gasoline from chainsawing on my hand, which is noxious substance. Well, it turns out that many, many noxious substances generate hormesis, and you're a little better off with some stress. Radiation, which was viewed to be bad, generates hormesis. And populations subject to radiation, maybe a small number of people have toxic doses of radiation and they die or get very sick. But a very large number of Hiroshima survivors or survivors of nuclear accidents in the world have less cancers, okay, because they've activated the stress pathways very well. So again, I'm trying to be honest here. I'll eat more plant-based supplements. I'll eat a terrific dinner, which is full of good foods, NRF2 activation, and in the bathroom I'll be cold again at night. So I think that the cold shock stress response is one I use to maintain my health. This is not conventional wisdom, these stress responses. Okay, now here's a very concise slide, which I'm sure you can completely immediately grasp about what you could do to keep your own stresses hormetic. Namely, there's a lot of things that I love this slide on it that have to do with stress management. But the main thing is to pursue things, to pursue stresses, allow stress. Oh yeah, the biggest source of stress I didn't talk about is doing this longevity science research. It drives me bat sometimes. I will wake up in the middle of the night and have to go on the computer and write something up, get a midnight insight. And so intellectual stress is a very important part about it, keeping an intellectually stressful life. Now, the pathways through which hormesis works, many of them are very, very well understood. I'm not going to go into them. These are all pathways that, molecular pathways for which drugs are being developed and may extend human lifespans. But on the whole, I personally don't think that drugs that address those pathways are going to particularly get us beyond the kind of wisdom of stress management, of dietary care, exercise, both the conventional and the unconventional wisdom. I think if you want the extra 10, 20 years, if you want to be one of those who lives longer instead of shorter, you could just elect to do that and study this stuff enough, begin to learn enough about it. I welcome you to my blog, www.agingsciences.com, and there are plenty of other sources. And it's not rocket science. It really isn't. It's really, I believe, within individuals' grasps. So I'm now going to wrap up with, well, what about breaking through the 122-year age limit? Well, the answer, if any, lies with innovative new solutions, new techniques. I mean, nobody's pulled this off despite searches for fountains of youth and other things. Nobody's been able to pull this off so far. So we have to look beyond everything. Certainly the field of molecular biology, of which there's a really impressive amount of activity going on here at the university, is an area that has to do with molecular pathways of activation. Certainly the field of epigenetics I've talked about will be a field. Hormesis, again, I'm going to say a word about that, and stem cell science. So now the hormesis pathways actually, you know, what lives longest? Well, we know that implying stress to certain organisms can extend their lifespans. So experimentally, we've done this with fruit flies and nematode worms called C. elegans. And one of the things you learned about very soon in studying longevity is about C. elegans, because they're a very favorite primitive mechanism. But some very long-lived species, the species that lives the longest, as far as we know, are the bristlecone pines that grow on the White Mountains in California. They grow at between 5,000 and 12,000 feet. And where they grow is kept secret because they don't want anybody cutting these things down. They're the oldest living things known to be in the world. Now, these beasties, these trees, you see some pictures of them, are subject to incredible stress. Variation of temperature of 70 or 80 degrees every day. And they go for months and months and months without moisture. Well, the pathways through which they deal with this stress are known, and they do it via hormesis. They've accommodated to living that long under conditions of very high stress. Again, another example I talked about is the naked mole rat. How many of you have naked mole rat pets? Okay, yeah. He and she live in tunnels underground. They are about the size of a mouse, but instead of living 2 years, they live up to 30 years. They live under horrible conditions: lack of oxygen, high carbon dioxide, acid environment, excrement, wars between colonies. They're very active. They're very nasty little beasties. But yet they live this long. Well, what's going on with them? Well, they actually show very little decline with aging. They maintain high activity, bone health, reproductive capacity. They don't get cancers, which is the main killers of mice. They don't get them. And they have incredible, they've been studied and have incredible stress management pathways. They do it. They've done it through hormetic accommodation somehow. They have for example, they have 6 times the amount of this substance NRF2 I was talking about that human beings have. Bats is another example. Again, compared to other animals of the same size, they live 20 to 40 years instead of 2 or 3 years, the size of a mouse again. And they, again, live under pretty horrible conditions. And they are subject to incredible stress, incredible oxidative stress. But yet they have very, very strong hormetic responses. So it raises the question of what role does the repeated stress play in these species being so long-lived? What has been the role of stress? And even further, or lack of stress. This is perhaps the course of evolution. And what role could stresses play, or hermetic stresses play, in extending life? I think it's an open question, human lives. So stem cell science is very important. How many of you know what stem cells are, or are familiar with them? Well, a major thrust, and also some work in this area is going on in this university. It's starting up now, having to do with creating what is called an induced pluripotent stem cell. Now, what does that mean? It means you can take a skin cell or a blood cell and you apply certain reversion factors, and those factors will transform that cell back into being similar to an embryonic stem cell. That means it becomes a cell that can differentiate into any other of several cell types. You can make any cell out of an embryonic stem cell, and they're very, very similar. Now, there's been a lot of work on perfecting this technique, increasing the productivity. It's about 7 or 8 years old. It's a very active area of science, and it's the basis of a whole field called regenerative mechanisms. Because, so again, the iPSC is functioning almost like an embryonic stem cell, and the technologies for producing these in high volumes, for producing them with high fidelity, for producing ones that really are pluripotent, that is, that really can differentiate into any cell. This work has been going on. It's a very active area of science. And they've been used experimentally in animals to regenerate organs. That is, you revert cells to induced pluripotent stem cells, and then you nudge them along to differentiate into being a certain cell type, like a heart cell, cardiomyocytes. Then you inject those cells or apply those cells, and in some cases there's been actual success in regenerating whole organs by doing that. So this is a field called regenerative medicine, and I think the places where they could be applied involve all kinds of diseases and all kinds of degenerative processes. So what's going on here? I have two final slides that say this. This is a normal process. A human being is conceived, and you have embryonic stem cells. You develop an embryo, and part of the embryonic development is that certain niches are formed in your body of what are called adult stem cells. These are stem cells. The embryonic stem cells can differentiate into every cell you have. How does it do it? It's a good act. The adult stem cells are not completely pluripotent, that is, they can't be, but these may generate into bloodline stem cells, mesenchymal stem cells. There's a number of more specialized stem cells or progenitor cells. So what happens when you live? Well, you have your normal body cells, your blood cells, they turn over. Every 120 days or so, all your blood cells turn over. They die. They reproduce, and they reproduce, and they reproduce, and they reproduce, reproductive cycle, and they die. But there are stem cells, adult stem cells, waiting. They have been reproducing too, and they are capable not only reproducing, but of differentiating. And they say, okay, we're going to make some new blood cells. They replace your blood cells. Almost all the cells we have in our body, even the very long-lived ones, eventually die or become senescent, and they're replaced by the adult stem cells. But what happens is that the adult stem cells get old and grumpy. They become what's called senescent. When they're old and grumpy, they can no longer differentiate into cells in a really effective way. They become senescent, which are these old neighbors. They don't, you know, they just throw the garbage out the window, and they affect other cells by them, cells that are proximate, and there's not enough reproduction to replace your body cells. Your organs get old, and you die. Okay, that's where we're at. And it's a once-through process, because once these embryonic stems, once your adult stem cells get old and grumpy, and they can no longer replace your somatic body cells, you're in the beginning of the process of beginning to have had it. Okay? All right. Now, what is a possibility? This is what I call the stem cell supply chain. Now, why? It's not advancing. Okay. Now, suppose that we could collect some somatic body cells, like skin cells or blood cells. Well, we can do that. We collect a small sample. Suppose that we snipped out any bad genes, like disease susceptibility genes, and snip in good ones in its place. Well, we know how to do that. We can do that. Suppose that we took those cells and we revert them back to PSC status. Well, we kind of know how to do that, okay? We kind of know how to do many of these steps. We replicate those iPSC cells. We kind of know how to do that, all right? We know a lot of things, actually. And then we induce them to differentiate into adult stem cells. Well, okay, we kind of know how to do that for many cell types. For many cell types, all you have to do is take a bunch of the induced pluripotent stem cells and just drop a cell of the type you want it to end up with, and it starts sending out signals. It says, hey, I'm a liver cell. Okay, let's all become liver cells. Okay, bunch, let's all become liver cells. And that's what happens. It's, we kind of know how to induce differentiation, but in this case, we don't want to induce differentiation into a heart cell or a liver cell. What we want to do is replace these key adult stem cells. We want to make these guys young again. So what we want to do is induce them to differentiate into adult stem cells, and then put those adult stem cells back in the little niches. They live in niches, like in bone marrow. Some live in tooth marrow. Some live in hair follicles. They live in the weirdest places, those stem cells. So we want them to renew them, make them young again, get these young cells, put them back in their niches, and then we have closed the loop. So that now it's not a once-through process, but there's a cycle that renews our cells. I think this ultimately, something like this, something along like this, which I call closing the loop in the stem cell supply chain. I think something like this is what might well get us beyond the 122-year lifespan. And there's been, I proposed this 2.5 years ago. Other people recognize the same thing. There's been a lot of progress since then. There's steady research. There are thousands of publications coming out of iPSC and organ renewal. And I expect to see something like this. Hopefully we could even get this all to go on within the body. Hopefully we could not even have to take the cells out to induce this kind of process to happen within the body. So, do I believe there's hope for breaking through the 122-year age limit? I do. I do. And I think if I had the list of candidates, I would say this together with studies of epigenetics, because the whole process of this cell reversion and inducing the differentiation, this is all has to do with epigenetics. I think these fields will give the possibility. So this is the essence of my talk. I will be putting a version of this presentation on my website, agingsciences.com. And when I do that, all of the links listed in red become hyperlinks, so you can do this. Actually, these are blog entries I've written on Hormesis, the NRF2 pathway, phytosubstances, and I've written a number of blog entries on this, extending the stem cell supply chain. So that's it.